Yeah, where am I? I am near, bacteria analogy. I think you need to keep somewhat, let me do it. It should be like this. Okay, fine. Listen here. So vegetative and dormant. Dormant I said seed. A simple example seed. And vegetative is plant. Okay. Now the bacteria can be in vegetative form. I will give example amoeba. Amoeba in vegetative form can be easily killed. We can able to kill amoeba in vegetative form. If amoeba enter into a dormant form, that means spore form, killing spores is very tough. Just like a seeds, plant seeds. Okay, coconut seed, you can see coconut how strong it is. Okay. So now this 0.1% is these seeds actually. Killing seeds is very tough. Spores. Now the same challenge we will encounter in the microbiology. We need to kill both the vegetative form plus dormant form. That will be the 100% killing. Okay. When we can able to kill 100%, then that process is called as sterilization. In sterilization, there will be 100% of killing both vegetative form and dormant form. That is called as sterilization. Then, what is this disinfection? Any ideas? How disinfection is different from sterilization? Yes, this is Mario. Yes, no. Yes, ma'am. 240 613 to 313 616. Different labs. I mean 616. I guess. Alright. What is disinfection? Any idea? What is the meaning of disinfection? Dis means something removing, infection means removing infection. So that is the meaning of disinfection. Now listen, disinfection means, for example, my son is 1 year old, so he will keep all mud in his mouth. Okay? Now, just keeping mud in the mouth will give you any infections. It also dependent on how much quantity of pathogens you are keeping in the mouth. Got it? 100 E. coli can cause a damage to my skin. But if I keep only 10 E. coli, that cannot damage my skin. You got my point? I was reducing the population to an extent where they won't cause any infection to me. 1 or 2 will be there, but they won't cause any infection. Reducing the infection can be stated as a disinfection. Okay, that is our Dettol. Okay, we are reducing the population to an extent where they won't cause any infection in the body. By the way, sterilization, I want to tell one important thing. In order to kill the seeds or spores of the bacteria, you need to keep the bacteria in a cooker. Okay, a very strong cooker, just like our kitchen cooker. And the temperature will be 121 degrees centigrade at 15 psi. Very high temperatures. In this temperature, we need to keep 15 minutes. Then we can kill all 100% bacteria. So we cannot do sterilization for a skin, right? I got a patient, he has a wound. Can I keep his hand in a cooker? No, he will become biryani there. So we cannot sterilize a human or living tissues. Then what approach we can take? We can go with disinfectants. Disinfectants are the reagents, chemicals that will reduce the pathogen count to an extent where they won't cause any infection. Some of the examples are Dettol, Lysol, etc. That is disinfectants. Clear, right? Now you understood the difference between disinfection and sterilization. Then what is this antiseptics? I'm sorry, I made a small mistake. A disinfectant cannot apply on living tissues, by the way. On living tissues, we cannot give disinfectants, okay? They are like strong acids. Disinfectants are used for cleaning surfaces, as she said somewhere, okay? Cleaning surfaces can be done by disinfection, okay? But we cannot use a for example, example, your Dettol or phenol. You cannot keep phenol on your skin. Okay. So yeah, there is a limitation to disinfection. If you can able to apply a disinfectant substance on a living tissue. When I say living tissue, there is a cut of tissue, there is a wound, and you need to apply some disinfectant there. That will be an antiseptic. If you can able to apply that on a living tissue, that will be an antiseptic. Can you give an example of antiseptic? Example of an antiseptic.
Sir, Vitadine.
Huh?
Vitadine.
Vitamin D?
Vitadine, sir.
Vitadine. Okay, penicillin.
Sir, it is a liquid form, brown color.
Okay.
Yeah.
Okay, fine. Got it. Yes, Vitadine. Huh?
Betadine is spirit.
Okay, spirit. Fine, Vitadine. I think. Yeah. Fine. Yes, or penicillin, Betadine, or Dettol, Savlon can be antiseptics. Okay. Then what is this antibiotics?
Penicillin.
Penicillin is an antibiotic. Very good. I will say very simply, antibiotics means antibiotics can also kill the microbes, and they are ingestible. You can even take them orally, or you can take into the system. They will be the antibiotics. All other things you cannot consume them, but antibiotics can be consumable. Okay, you can ingest them. Okay, so these are the various types of methods of killing microbes. Okay, so now we are focusing on sterilization in microbiology. Why do we need to sterilize, by the way? What is the purpose of sterilization?
Kill all bacteria.
You kill everything. Why we are killing everything when we are studying them in the first place?
Yes, sir.
Sir, so that it could not react with the external environment and the microbes in the external
She's
environment.
saying sterilization because it cannot react with the external environment. Justify?
Yes, sir.
Okay, listen. Microbes are everywhere, right? So I'm going to make a microbe favorite substance that is called as culture media actually. Okay, that is the biryani to a microbe, to a bacteria. Okay, so I made a culture media. I kept culture media on this table. Okay, why I made culture media? Because I want to study an infection in a patient. I took patient sample, okay, and I kept this sample in this media. So the pathogen or the microbe in the sample is not growing in the petri dish. That is the idea of making petri, medias. Now, there is a chance of getting contamination. What is contamination means? When I prepare this media, when I am talking here, some of my saliva particles might enter, on this dish, and that, everywhere microbes, even in my saliva, they will outgrow, and we will think that patient has some infection. Since we collected a sample, it is a false positive. Patient don't have any infection, but I got a infection growth, some growth because of my contamination. To remove such contamination issues, we need to sterilize the media. The media should be clean. How we will ensure the media is clean or not? We are going with sterilization. First we will sterilize the media and we will keep in this laminar hood. In that hood only we will inoculate the sample. Okay? If you are doing that inoculation outside, what happens? There is a cross contamination. So that's why we are doing sterilization. So what are the methods of sterilizing? How we can kill microbes? How we can kill microbes? Yeah. Methods of killing microbes. Think realistically. Yes. How we can kill microbes? Can I torture a microbe by keeping fire? Microbe on fire? We can, right? Yes. Yes. Yes, Shyam.
Autoclave में डाल के high temperature.
Autoclave, he already have one answer here, so he is confident. Yes, autoclave will kill microbes anymore.
Hot air oven.
Hot air oven, very good. Hot air oven will kill microbes. Very good. What is the difference between autoclave and hot air oven then?
So
Hot air.
Oh, very good. Hot air oven has a dry heat and autoclave have moist heat. What is a dry heat and moist heat, by the way?
अब बोल.
The answer is in the word only. Dry heat means directly you are exposing to some heating agent. Okay, that is a dry heat, and moist heat will be under some moisture, some water. Give me some examples of dry heat sterilization. Yeah. Methods of dry heating. Why are you confusing? Why are you afraid? Listen, there is a candle and there is an open fire. Can I keep microbe on an open fire? That will be dry heat. Okay? With a loop, heating loop, we will heat the loop. Okay, that is a dry heat sterilization. So open flame is there. Any other such a dry heat sterilizations?
So
Can I keep a petri dish on the sunlight?
yes sir.
So sunlight can also kill the microbes, right? So yes, you are eating dry fish, right? So that they are, technically it is sterilization. Next, sunlight we can use any more?
Hot plate. Hot plate.
Hot plate, very good. Hot plate to an extent, but we'll keep it aside. Next, direct heat. Open flame we discussed, then sunlight we discussed. Any more? Hot air oven. Hot air will be circulated through the sample. So that is a dry heat. Any other such torturing things to the microbes? Can I use microwaves? No, microwaves will boil inside water and they will. So microwaving can be done. Can I use UV light? Irradiation. I can use UV light and I can kill microbes. Any other methods? Dry heats. Radiation I discussed. Then UV, radiation, sunlight, open flame, then microwaving. Any more other torturing things to the microbes? Can I filter? Can I use filters to sterilize microbes? Is it possible to use filters? A very advanced filters. Filters that they can even filtrate nanoparticles. Actually, filtration is possible, okay? HPLC, high performance liquid chromatography. There are some filters. Or N96 mask.
Yes sir.
N96 mask is a filter that can even block viruses. Viruses are the smallest living creatures, right? So filtrations, we can do filters to
N95.
achieve sterilization. Yes.
N95 is
N95.
N95.
Then moist heat sterilization. Coming to the moist heat, we are using some water or moist containing sterilizing approaches. Now give me examples. One is your chawal dal, that cooker. Okay, that is autoclave. The technical term for cooker is autoclave in medical terms. Okay. Then any more? Moist heat sterilization apart from autoclave. Give me examples. Moist heat apart from autoclave. Any idea? How you said you know autoclave, right? Then you should know others also. Other moist heat techniques. You know or you don't know? Don't know. Okay, then listen. Boiling. Take a water, boiling water, suspend the article. It will kill the microbes, okay? There is one person, for his lifetime he worked on it, on moist heat sterilization, and you are not recognizing him. That is very sad to me. Okay? This person said, if this is causing, you just boil it and it will be sterilized. And you just feel like this. Since he observed this one, so he kept his own name for the process. Like if I discover something that might be Vamsization, something, okay? And that is pasteurization. Louis Pasteur.
Yes, sir.
Okay sir.
What is pasteurization? Tell me, students. What is pasteurization? So just now you said something.
Hot temperature पे वो करते हैं ना, filter करते हैं.
See, milk will be boiled at certain degree centigrade, okay, 42 to 46 for half an hour. So that will kill all lactobacillus or similar pathogens in the milk. So that is called pasteurization. So pasteurization is a moist heat sterilization. Any more? Now there are some problems with moist heat. Okay, like autoclaving we discussed. Autoclaving is a cooker and we are keeping articles in a cooker. Can we able to keep everything in autoclave? Is it possible?
No, sir.
Like I need to autoclave a catheter, a plastic tube. Can I keep that plastic tube in a cooker?
No.
I cannot. You know that plastic will be melted. So how can I sterilize a plastic tube? You know, catheters are very costly, 5 to 6,000. Simple pipe, you know, you need to keep in the urethra, in the bladder, very very costly. So they will go for sterilization sometimes. That depends. Yes, tell me, how to sterilize a plastic material? Can I keep it on an open flame?
No, sir.
No. Then what should I do? And that is where we are having different approaches. Okay. Is it possible to sterilize this entire room? Every whole room should be sterilized. Is it possible?
No, sir.
Not possible, right? Yes. Now what we will do is we will make a very toxic fumes. Toxic fumes such as formaldehyde. Formaldehyde is a gas. So we will make some reaction. Formaldehyde gas will be, vapors will be released, and that vapor we will keep this room in an enclosed area. Okay, we will close all the doors and we will release the vapors. Formaldehyde can able to kill microbes. Okay, to an extent. So that technique is called fumigation. Okay, yeah, fumigation. Here we are using formaldehyde to fumigate rooms, large areas. Then any other interesting things, you know?
So
How
what
many
is-
know? Yes. The answer. Okay. There is one more technique called inspissation. What is inspissation? Any idea? Yeah, inspissation also. Similarly, we will keep the sample on a vapor for half an hour. Okay, that is inspissation. I hope you got some idea about the sterilization and all these things, right?
So what is fumigation?
Fumigation, subjecting to a gaseous vapors, is called fumigation. Subjecting to gaseous vapors. Okay. I will give one example of fumigation. What example I can give? Simple fumigation. Have you smelled formaldehyde? No, right? Formalin. You don't know. Do you know what is the meaning of volatile substances? Volatile substances. If okay, I will tell even simply. If there is a reagent in a bottle, you open the bottle and you are getting a very strong smell. They are volatile substances. That means they can easily spread in the environment. Okay, so they are called volatile substances. So what I will do is I will take a formaldehyde, a very very volatile gas. I will dissolve this gas into a water and I will make it to come fuming, fumes. Do you know nebulizer? How nebulizer works? The children nebulizer,
Yes.
they will get that fumes, right? Similarly, we will try to convert formaldehyde into that fumes, and that fume. Okay, I got the answer. Fog. We will convert that substance into a fog with some machinery, and that fog will be subjected to one particular area, and that process is called fumigation. Fog fumigation. Okay.
Like diffusion?
Huh?
Diffusion.
Okay, I got the answer. Wait, I got the answer. I'm becoming more creative. How many of you see on stages there will be the black smoke on stages? That is a fumigator actually. But it has only water vapor. It will give water vapor, okay? So when you are dancing, you will get some vapor, smoke, right? Actually, that is water vapor, and that process is called fumigation. Conversion of liquid to some vapors. So here we are using some toxic fumes to kill the microbes. Yeah, that is the idea behind fumigation. Any more doubts so far? Fumigation. Yeah. So, can you tell me the temperature of autoclave now? I already wrote on board. What is the temperature of autoclave?
121.
121 degrees centigrade. Okay. What is the pressure of autoclave? I wrote it on board. See.
50 psi.
50 psi, 15 psi,
15
15 psi. And what is the time or duration of autoclaving?
15 minutes.
15
For 15 minutes. So this is the gold standard timing for autoclaving. That is at 121 degrees centigrade at 15 psi for 15 minutes is the autoclaving temperature. Got it? What is the temperature of hot air oven?
220.
140.
140.
160
Really?
degrees Celsius.
Wrong. 140 is wrong.
160.
160 at what time? Duration? Okay, 160 1 hour, then 170 half an hour, 185 minutes. Okay, the higher the temperature, the decrease the temperature time. Okay, that is hot air oven. Then yeah, in what criteria you will hot air oven rather than autoclaving? What substances you will subject to hot air oven? What things you will sterilize in hot air oven? They are watching at hot air oven, like there is some answer there. Yes, tell me students.
So, test tube beakers and all.
Yes. Okay, empty test tubes, empty beakers can be glassware, can be sterilized with hot air oven. Any other things you can sterilize with hot air oven? Glassware, then?
China dish.
Hmm?
China dish.
China dish.
Sir, if
Okay, fine. Then?
All glasswares.
Bonus
Glassware.
glassware already done anymore? Hot
Measurement
air oven?
tube. Measurement tube.
Measurement tubes, that is also glassware. Then, just giving names of glassware now. See, smart. Can I sterilize metals in the hot air oven, like blades, scalpels, forceps? They can be sterilizable. Okay. Then, in what condition I will use autoclave? What articles will be autoclaved? Medias, then
samples.
plastic-ware, tolerable plastic-ware, autoclavable plastic-ware. Then, can I keep apron in hot air oven? Can I? Can I sterilize apron in hot air oven? I cannot. Can I autoclave an apron?
No.
Some of the PPE are autoclavable. Gloves, autoclaving gloves and then autoclaving masks are there. Okay, yes. Some things can be autoclavable. Anyone? What things I will go for open flaming then? Direct heat subjection, direct open fire. What articles I will sterilize with open flame? Inoculating loop. I hope you studied what is inoculating loop, right? How you will sterilize inoculating loop? By keeping it on an open flame, right? So this is all about sterilizing. So in sterilization you need to understand what are the various methods of sterilization, what items can be sterilized in what methods you need to understand. Okay? So this is all about sterilization. Thank you very much.
Thank you.
Thanks.
You can leave students.
Thanks.